Most of you that have studied telomeres and read about them in the newspapers have probably heard that telomeres are like the caps on our shoelaces, that when they wear away, they wear away, they get frayed, and that's exactly what happens to our telomeres. They get frayed away just like the caps on our shoelaces do. Now, I don't like that example, okay? I mention it only because it's so well publicized, but the principal part of telomere shortening has nothing to do with wear and tear. It's a common misconception about telomere biology. What's really going on is a more passive kind of phenomena called the in-replication problem. It's more of a lack of a function than something that's causing the degradation. What I'd like to do is run through DNA replication using an analogy. What I'd like to do is I'd like to compare DNA replication to the making of a brick wall. Okay, so you have look at these bricks here. Imagine that's the top layer of a brick wall. When a brick layer goes and makes the next layer, that's the same as replicating the DNA. And we know every time our cells divide, our chromosomes have to replicate. So we have a brick layer that comes along, and he's laying bricks. And remember that our chromosomes are like 10 million bases long, so this is a long, long process. And so the person's got a long ways to go. There's origins of replications about every 10,000 to 100,000 bases. But what we want to really do right here is see what happens when we get to the very end of the chromosome. Okay, so here we scroll over to the telomere, and you can see when he gets here that he's going to have a little bit of a problem when he gets there. And this is exactly what happens inside the cell. He cannot replicate the very end of the DNA, the telomere. And so as a result, the new chromosome is shorter. And this happens cell division after cell division. But no matter what you do, no matter how well you eat, no matter how much you exercise, and no matter how much you do everything your doctor tells you to do, there's absolutely nothing you can do about this. These telomeres are going to shorten like this. Now, I did say that it's not a wear and tear phenomena, but wear and tear, I just said it wasn't principally a wear and tear phenomena. Wear and tear does occur. You can actually accelerate your telomere shortening by leading an unhealthy lifestyle. Examples are anything that would cause stress, and those kind of things are going to cause free radicals, and free radicals can induce damage. And smoking is a good example. Smoking causes free radicals, and that can accelerate your telomere shortening. So the answer is, statement is don't smoke. Don't do anything. And here you can. Exercise helps, eating right helps, and doing whatever your anti-aging doctor says to do, do it, because it will relieve stress. So telomere shortening has been implicated in not just aging, but in a lot of aspects of normal aging. And I'm just going to list a bunch of them and describe them for a few seconds each. Skin aging. We lose skin cells all the time. We're sloughing off skin cells. When those cells are lost, other cells deeper inside, the stem cells particularly, have to divide to replace those cells. As a result, the telomeres get shorter. And so that after many years, your telomeres have gotten very short, your skin cells senesce, and that's why your skin starts looking old. Let's say that's one of the reasons why your skin cells start looking old. Atherosclerosis. We have shown in numerous experiments now that when we take endothelial cells, aligning the blood vessels where there's plaque, the telomeres in those endothelial cells are shorter than endothelial cells found elsewhere in the blood vessels. The same is true for the smooth muscle cells underlying those endothelial cells. But again, especially where you have turbulence in your blood, you're going to be stripping away endothelial cells. Other cells have to divide to replace those cells. As a result, your telomeres get shorter. And when the telomeres get short, cells get unhealthy, and they get a decreased ability to prevent plaque buildup. The common rule of thumb here is bad things happen when telomeres get short. So we want to keep them long. Another one is osteoarthritis. Our joints are covered with chondrocyte cells. Overuse of those joints can cause those chondrocytes to wear away. That's okay at first because you have cells that can divide to replace those cells. But after a while, the telomeres get short, and you can't replace your chondrocyte cells, you get bone on bone, you have osteoarthritis. All these things I'm talking about, if we could find a drug or some treatment that could prevent telomere shortening or reverse telomere shortening, could be a cure for some of these things. Osteoporosis, we have the osteoclasts and the osteoblasts. The osteoclasts are tearing down the bone, the osteoblasts are rebuilding it. Well, the osteoclasts are more dependent on cell division than the osteoclasts. And as a result, telomeres can get shorter. So again, this can result in the osteoblast becoming senescent and lead to the osteoclast dominating, making your bones weak, and is one of the possible causes of osteoporosis. And I got to remember, I'm a research scientist, so most of what I'm talking about is still in the theory stage. We probably won't ever be able to prove some of these things until we actually have a drug that prevents telomere shortening. General immunity. Our immune cells, whenever we have to fight a disease or infection of anything, immune cells have to divide. Telomeres get short. As a result, we eventually suffer from immune senescence. Alzheimer's disease. Alzheimer's disease, could be due to beta amyloid plaques or the tangles, but it's also possible it's due to weakened immune systems. There's a lot of papers suggesting that the telomeres and the astrocytes get shorter in Alzheimer's patients, and it has been shown that just regular blood lymphocytes, the telomeres in those cells get shorter in Alzheimer's patients too. Though there's still no question, nobody knows anything about cause and effect there. Macular degeneration, #1 cause of blindness in the elderly. We have retinopigmental epithelial cells in the back lining of our eyes. When those cells start getting short telomeres, these cells senesce, and that causes macular degeneration. Again, if we could find a drug that prevented telomere shortening, that could possibly be a cure for macular degeneration, if it was the only thing that caused macular degeneration. Liver cirrhosis, I can go on and on, but you're killing your liver cells, other cells have to divide to replace them. Again, telomeres get short. Muscular dystrophy, I can make the same kind of argument. AIDS, the AIDS virus infects the CD4 cells. CD8 cells have to fight unsuccessfully to fight the virus, and eventually the telomeres run out on the CD8 cells, the cells senescent, and people are saying, well, AIDS patients have no T cells. It's because they senesced away from telomere shortening. Not added here is cancer. Cancer is also something that telomere length, even though I've worked a lot on trying to inhibit telomeres to kill cancers, keeping your telomeres long might be a good way to prevent getting cancer. Keeping your immune system strong, and I was saying before that bad things happen when telomeres get short. When your telomeres get short, that causes a lot of rearrangements, mutations in your chromosome, which can cause a lot of mutations that lead to cancer. So keeping telomeres long is also a possible way to decrease the risk of getting cancer. Okay, so other victims of telomere shortening, just research in the lab, scientists working in the lab are always plagued with the fact that they get endothelial cells, let's say, from somebody's blood vessels, and they can only divide a certain number of times, and then they have to get more because they can't do their experiment anymore. So having a drug that they could use would be useful. Monoclonal antibodies, I'm going to skip that one for now. Skin transplants. Well, let's say skin transplants and bone marrow transplants both are plagued with telomere shortening problems. You take the cells out of a person, you expand them in the lab, the telomeres get shorter when you expand them. You put them back in, the cells are older than the person, so at least cellularly older. And the new stem cell technology, that's really going to be plagued with telomere shortening problems, except for the human embryonic stem cells, which don't have a telomere shortening problem. All the rest of the adult stem cells do have telomere shortening. And so whenever anybody is going to take stem cells from a person, expand them, put them back in, just like we were talking about the bone marrow transplants, which is essentially the same thing, we're going to have telomere shortening. So I think the telomere stem cell technology field is going to benefit tremendously from some type of drug that it prevents telomere shortening. This is just a list of all of them, and the key thing is there are a lot of them. Learning about telomere biology and controlling telomere biology is going to offer opportunities in lots of different places in the medical field. Any place in the human body where we have cell division is going to be a place where there can be a disease that can be affected by telomeres. So, I would like to say that I believe telomere shortening is a disease, and diseases need to be cured. Okay, so we've said that the green cells don't have any telomere shortening, the orange ones do. Now we can say, let's see, so I guess okay, I'm sorry. So the question now is, why? So if we want to cure this telomere shortening, we got to first understand why it doesn't happen in our germline cells. And that introduces another word that I suspect will be a household word in 5 to 10 years, and that's telomerase. And telomerase is an enzyme that is like a factory inside of our cells. The gray part of this cartoon is showing the telomerase protein component, the enzyme, and it essentially binds to the very tips of the chromosomes. What's shown in green is the telomeres, the tips of the chromosome. Here it now shown as a double helix. And the telomerase adds nucleotides to the end of the DNA to lengthen the telomere. So we discovered telomerase on July 6, 1994, and I led this research group. And as we usually do as research scientists, we go out and celebrate with pizza and beer, and had the pizza delivery guy take a picture of us. But the guy in the blue shirt, that's me, and I want to point out 3 other people who I ended up sharing this National Inventor of the Year award with for this discovery. That's Brian Villapanto on my left in the white shirt, Walter Funk in the purple shirt on the right, and Junlin Fong, who's back in the red shirt. You can barely see her there. But I will be talking about Walter Funk again in a few minutes. Okay, so now, what is going on with telomerase? The bricklayer is still just as unable to replicate the very ends of the chromosomes as he was before. But now, like an angel, telomerase comes in and replaces that missing brick. And so that's essentially, in a concept level, that's what happens inside the cell. So every time the DNA replicates, the chromosomes do not get shorter. How do we know this? When we first discovered telomerase, we couldn't know this before discovering telomerase, but once we did discover telomerase, we did a whole series of experiments at Geron Corporation. As I said before, human cells senesce. Okay, let's say normal human cells senesce. They start, when you put them in a petri dish, they'll divide fine, and then all of a sudden start slowing down, level off, and we have senescence. When we now we have the telomerase gene, now we put the telomerase gene into these cells, and we call this telomerizing them. So when you see the word telomerize cells or telomerizing, that means we put the gene into the cells. It's now a very published term when you talk about telomerase biology. But when we did that, we found the cells did not start slowing down, did not have any senescence, and more importantly, they were not cancer. This is like you'd expect a cancer cell to grow like this. These cells were not cancer, and a lot of literature since this time has shown that they actually have a decreased risk of becoming cancer. So this was the absolute first proof. Before I said we didn't know cause and effect. Does aging cause telomere shortening, or does telomere shortening cause aging, or more specifically, organismal aging? We can now say that telomeres are the cause of replicative senescence. So we published this in 1998 in Science. This made headline news in New York Times, called it the fountain of youth. A little premature, but it did make a lot of news. Sometimes this doesn't react fast enough. Okay, we measured the telomeres in these cells, and we did see that they stopped shortening, but they did more than that. They actually got longer. And this was a real surprise to us in 1998, and it begged the question, did the cells actually get younger? And so it was about this time that I left Geron Corporation and went and started my own company, Sierra Sciences. Even though we now are different companies, we still work very close together. But Geron still continues some of the work, and Walter Funk, the guy in the purple shirt that I showed you earlier, did some experiment where he took young and old human cells, and he put the telomerase gene into the old cells, and he also had some old cells that he didn't put the telomerase gene into. And then he cultured these cells into skin on the back of a mouse. Now, you couldn't do this with a human because the FDA would never allow it, but you can do this with mice. So he looked at the young skin, the skin from the young cells growing on the back of the mouse, and they looked young. When he looked at the cells from the skin from the old cells, the skin looked old.